Last talk of the session. And it's for me, again, a pleasure to introduce you Pascual Sánchez-Juan. Yes. Which is the scientific director of the Fundación CIEN and also a member of CIBERNED. He's going to talk about plasma biomarkers in the Vallecas study, bridging the gap on the road to the clinic.
Thank you very much, Miguel. Well, I'm lucky that I just talk after Alberto because I'm going to be explaining some of the work we've been doing with plasma biomarkers determined by CIEN in our course. Basically, in the court is leading Alberto, which is quite a unique resource. And first, I have to apologize because we do not have consolidated data. And this is a preliminary analysis, but we thought that it was worth it to show it to you because we want you to know what we are doing and the potential and the great potential of our study, and to give you the message that we are very open to collaborate with CIBERNED. So I would like to start with some bad news from last week. One is the trial of an anti-tau that was published last week, showing that semorinimab, an anti-tau against the N-terminal part of tau, it didn't work in Alzheimer's disease. Luc Bouet was talking about it yesterday. And also very bad news from the amyloid field as well. Even worse, in the Colombian study, the anti-amyloid drug, the crenezumab from Roche, didn't work also after a long study. So very, very disappointing. I think to me the message of this is that, well, several messages, of course. First, we need more basic research. We need to understand what is going on. That is essential. Probably thinking as a clinician, we're going to need to combine several treatments. This is probably, there's not going to be a magic bullet for sure. And probably we have to look beyond amyloid, beyond tau. We have to really look for other pathways. And this is a trend in this, the Cummings report that he released every year of the pipeline of clinical trials ongoing on Alzheimer's disease. We see a trend in changing, so more alternative pathways are on clinical trials in addition to amyloid, to tau. We find more alternatives in clinical research, but also we find this trend in preclinical research even stronger. So we see now that, for example, neuroinflammatory pathways are being tested in second place after amyloid. So this is a change. And another trend that we are finding is that many targets are based on genetic risk variant that has been discovered by GWAS. This is kind of guiding the way to look for alternative targets. As an example, you have here the analysis on antibodies against TREM2, modulating TREM2 are ongoing in Alzheimer's disease, and of course, Bruce Miller and Raquel also talked yesterday about the antibodies against sortilin-1 trying to increase progranulin in frontotemporal dementia and Alzheimer's disease patients. So I think these are exciting developments, but definitely, again, as a clinician, I think most of us agree that if a disease-modifying therapy is going to be available, it has to start as soon as possible. So we probably, ideally, we should diagnose not the patient, but the risk of an individual to develop the disease. I think this is going to be an essential point if we want that something works. Probably, most surely, we will need to combine several drugs. It seems that it's not going to be enough with only one. And most likely, we will need to tailor the treatment for the patient. So probably this is going to be an important thing. And to do that, we definitely, clinicians, we need to do a better job. We need to diagnose the patients before, or as I said before, even when they are not patients. So that would be ideal. We need to categorize risk in preclinical disease. And we need to we need a precise determination of these pathological components that Alberto was showing. All these bars are components that we may need to understand before treating the patient. And very, very importantly also, once we have the patient, we need to monitor the drug response for each pathological component. And this is difficult. This is the same slide that I stole from Alberto. All this complexity that you find in the brains of our patients. We clinicians, when we see the patient, we see the syndrome, but within the syndrome, we find Alzheimer's disease, but we found many other things, as Alberto showed before. So that leads us to the importance of biomarkers in clinical work. We definitely need biomarkers if we want to go one step further, thinking of course, in having an available treatment. So we really need to go further in the diagnosis, and we have to consider all this complexity when we do the diagnosis and we treat the patients. Not only clinicians, but of course also basic researchers. You have to also consider all this complexity when you generate the models of the disease, which is much more difficult, at least as we clinicians expected. And definitely we need biomarkers. If we want to be so accurate and do this early diagnosis, we have not enough with our clinical tools. And well, CSF and PET biomarkers are consolidated, are great biomarkers for Alzheimer's disease, at least for detecting tau and amyloid. But they definitely are not enough if we really want to do to scale the diagnosis and to make it more simple and democratic. So probably we need new techniques like Alberto was saying before, like the plasma biomarkers. So luckily, Inga was talking yesterday about it. There's been an explosion of research analyzing these blood biomarkers with different techniques. I will focus on CIMOA, is what we have in Fundación CIEN, but there are different techniques, and they are very consistent, the results of these techniques, and they allow us to determine with a lot of fidelity, if you compare to the CSF, the ATN scale. So we can determine in blood now amyloid, we can determine different species of tau. Yesterday, it was mentioned, the 181, the 217, and the 231 for different phases of the disease. Also neurodegeneration biomarkers like NFL, and also very recently, astroglial markers. Very interesting, GFAP, we will talk about it later. So, out of all these techniques that are being used to determine plasma biomarkers, probably CIMOA is the one that is closer to the clinical application. Luckily, new techniques are coming, so this is going to be very good because prices hopefully will decrease because now they are really, really prohibitive. But of course, in this road, as the title of my presentation, this road towards the clinical implementation is what we want as a clinician, but we still have some problems, some gaps that well, there are many more, but I just mentioned here the one that we are focusing on to address, we are trying to address in Fundación CIEN. Well, first of all, most of the data that we have come from selected cohorts, like ADNI, for example. These are very selected individuals without co-pathology, with some criteria, stringent criteria. So we think that there is a need of real-world data, so something more close to what we find in the clinic. Very, very importantly, there are not many studies addressing what happened in late-stage dementia. We don't know what's the behavior of these biomarkers at the end of the disease. We don't know what is the diagnostic performance, these stages, and we don't know what is the trajectory, how they change through time. And very importantly, there is a scarce correlation between plasma biomarkers and the neuropathology. There are not that many studies assessing this, which is a fundamental step. If you want to implement this in the clinic, you need the neuropathological confirmation. Most of the studies correlate plasma biomarkers with CSF and with PET, but not with neuropathology, and this is an important step. And this question has to be answered, how the plasma biomarkers reflect the intensity of the pathology change, this ABC score that Alberto was saying, how it reflects the changing, how it reflects the progression of the disease, and how they reflect the non-Alzheimer's disease pathology, this mixed pathology, co-pathology, whatever we call it, how they are, how is related to the peripheral levels of plasma biomarkers. This is an important question that we, definitely clinicians, need to know. And well, this busy table is just to give you an idea of the just 12 studies that are addressing these important topics. So I reviewed the literature, I reviewed the PubMed, and I've only found 12 studies that are correlating plasma biomarkers and neuropathology. And as you can see, in the bottom, you have, probably it's very small, but this is our project, the Vallecas Alzheimer project. Most of the project has a small n, lower than 100. As Alberto said, we have 137 brains with the plasma samples, which is one of the largest samples. Most of the studies do not have longitudinal data, it's just cross-sectional, plasma and brain. We have, we have follow-up data, as Alberto just explained. Most of the studies show data for only one biomarkers or two biomarkers. Only three has a panel of biomarkers, as we are doing. We are analyzing all the Alzheimer's disease commercial plasma biomarkers for CIMOA. And only 3 studies has analyzed mixed pathology in addition to our sample. And I think, to me, the biggest advantage that we have is the time due to the unique setting of our study, because we are within the nursing home, and the brain bank of Alberto is within the nursing home. So we get the patients immediately and as they die. So the average time from the plasma extraction, as Alberto says, every 6 months, from the plasma extraction to the autopsy is only 141 days. So it's very short, the time between plasma extraction and when we analyze the brain. And as you can see, well, probably you cannot see, but I tell you, in most of the studies, it's more than 2 years, 5 years, 4 years, so it's a long distance. So our data is much more accurate to any of the data that has been published. Oh, sorry. So I'm not going to repeat this because Brian yesterday described the Vallecas project and Alberto just described the Alzheimer project. These are our cohort, and we want to use this cohort to address some of these questions. I think, well, I will skip this because you already know. I'm not going to repeat. I'm not going to enter into that because these are all neuropathological variables that Alberto is measuring. So as you can see, it's very comprehensive, and he has a very detailed information of all the co-pathology in addition to Alzheimer pathology. And this is our population. We have, of course, all the Vallecas Alzheimer patients in the study. They are 137, and we selected from the Vallecas cohort controls. We selected 133 controls that have been cognitively stable for almost 10 years. So they are very good controls against neuropathologically confirmed Alzheimer's disease cases. Alberto mentioned the demographics. Just to show you that there are no big difference between age, 83.5 in the Alzheimer study, and in the Vallecas 82. They are only 1 year younger in the Vallecas. Of course, the functional staging is very advanced in the residency. They are all moderate to severe dementia, most of them, and in the Vallecas they are independent persons without any functional problems. And now we enter in trying to answer this question that I mentioned before. The first one is, what is the diagnostic performance in this population? How well performed the test in this population? So we tested all these biomarkers, A-beta 40-40, total tau, phospho-tau 181. We do not have completed the information for 181, so the data I'm going to present is not final. We have GFAP and NFL. And what we want to study is how they perform comparing these late stage dementia neuropathologically confirmed cases compared to the Vallecas cohort. And on average, the time from the first sample, because we use the first sample when they have a similar age, both cohorts, to death is 5 years. So, and these are the ROC curves. We use this analysis to have an idea of the performance of the test. And as you can see, we have a very, very good performance of all these tests to discriminate between both populations. If you look at here, the amyloid test, the A-beta 4240 has a 0.93, which is an excellent area under the curve to discriminate between these two populations. The neurodegenerative markers also show excellent properties, the GFAP and the NFL, more than 90%, and the total tau is also quite good, 0.87, and as I mentioned to you, the phospho-tau is not complete, so you better not pay attention to this. So that was performance. The second objective is the trajectory, how they change with time. And again, we now focus only in the Alzheimer project, so pathologically confirmed population of late stage dementia, and we analyze 3 samples, 3 consecutive samples to see the changes in time. This is data for GFAP, probably is very small, but I explained this graph because I'm going to repeat it. In this axis, we see the levels of the biomarkers, and these are the groups. These are the controls. They are in the same scales with the patients. This is the baseline, the second blood sample, and this is the pre-mortem sample. On average, there is 2 years between these first 2 samples and 3 years between the middle one and the last. And between the last one, the pre-mortem and death, is only 141 days, as I said to you. So very short period. And well, first you have to look at the difference between cases and controls, which is quite high. You cannot see from there, but it's almost 120% more. So it's very good for diagnosis, a big range. And you see that there is also an increment, a linear increment that is significant, very significant in the linear mix model. So it's telling us that it's a linear increase in time of almost 44 to 41% in every sample. So it's also seems to be not only good for diagnosis, but also for follow-up, because we have quite a linear increase during the disease course. And now these are neurodegeneration markers. First, we have total tau. In total tau, we have a different behavior, as you can see. The difference between controls and cases is substantial. So we have, I mean, it is concordant with the fact that we have good ROC and area under the curve. But what we find here is that the trajectory is quite flat. It increased a little bit and decreased later on. And in the linear mix model, it's not significant. We do not find a linear increase in total tau, which is concordant to previous data that suggests that it plateaus at the end of the disease. In NFL, we have different results, very surprising. We find first that kind of also a plateau. Well, the difference with controls is huge also, so it's, the area under the curve is very high also for NFL, as I showed before. And what we find is a very sharp increase in the last sample. I have not an explanation for that. We really have to analyze better the data. But I have my hypothesis, of course, of why this is happening, which is so substantial. But maybe in the question time we can talk about it, and I would love to know your opinion about that. In both cases, the linear mix model is not significant, so we do not see a linear increase in both of these neuropathology biomarkers. And now the amyloid biomarkers, A-beta 40, no good for diagnosis. You see no difference between cases and controls. And even though we see a linear increase that is significant in the linear mix model, is very low. The beta is very low, so it's almost flat. But A-beta 40 increase, but in a very small fashion. A-beta 42, very interesting, very good for diagnosis. We see that controls has much higher levels than cases, good area under the curve. And we see, surprisingly, a linear trend increasing in time. Again, very small beta, but significant in the linear mix model. So beta 42 in our population increased with time. Again, I have my hypothesis, but maybe we can discuss later why this is happening, because we should expect the opposite, right? And well, similarly happened with the A-beta 42/40, the ratio. very good for diagnosis. You see a big difference between cases and controls, area under the curve, 93%. And we see a very, very light increase in time, is just 3% in every sample, which is significant in the linear mixed model, but is insignificant. And finally, tau markers. As I said to you, this data set is not completed. We ran out of kits, of Simoa kits, so we have to wait for it. But it seems that it's a good diagnostic marker. We see difference, and it seems to have a linear trend, but we really have to finish because these are just a very small sample. Okay, now objective 3. Very important to correlate the plasma biomarkers with the neuropathological findings. First, we focus on the pathological stages, this A, B, C that Alberto was mentioning for amyloid, for tau, how the disease progressed neuropathologically, how this correlate with the, and now we are going to focus on the pre-mortem sample, the one that is so close to the brain extraction, only 141 days. So the first thing that we study is to correlate with brain weight. Just it's a gross measure of disease progression, but it's quantitative, and we found a correlation with GFAP and the brain weight. Even adjusting for covariates, we found that this correlation was quite robust with GFAP, and we didn't find this correlation with brain weight with the other markers. I think very interesting finding. Still, we don't have an explanation for that, but that's something that really we have to go deeper. Again, in the macro study of the brain, we also correlate with the pattern of atrophy, and we found again association with GFAP and temporal lobe atrophy. And again, that was only GFAP, the only biomarker that correlated with this temporal atrophy pattern that Alberto measured semi-quantitatively. So we saw that GFAP was associated not only to brain atrophy, but also to temporal lobe atrophy specifically. And that association was not found for any of the other markers. Okay, and now we started to correlate with the ABC stage scheme that Alberto mentioned, and this are the correlations with GFAP. GFAP correlates with all the ABC parameters. The A is the TAL, so it's the distribution of A beta plaques, and is correlated in the linear model, adjusted by covariates. We also found the strongest correlation was with the NIA-B, which is the equivalent of BRAC, is tau. A very significant correlation between GFAP and the tau staging. And also correlates with the NIA-C, which is the CIRAD score, which it will translate at the intensity of the neuritic plaques. And GFAP also correlates with this stage. And this is the ABC score that Alberto was mentioning. When you sum up all these ABC, you get this score. And also we found a correlation between GFAP and the composite score, including all the ABC. So it seems that GFAP is really good correlating to all the stages of the AD pathology. We do the same for the rest of the markers, and we found that for the ratio A beta 40/40, the only correlation was with the C, the NIA-C, which, as I mentioned to you, is the CIRAD score, is the intensity of the fibrillary plaque. So it's the only one that correlates with A beta 40/40, and so this is, I think, concordant with the fact that the amyloid markers are not good to follow up. They are not that good tracking the disease progression, and the A beta 40 and A beta 42 didn't correlate with any of the other scales. And finally, the phospho-tau 181. For this analysis, for the promortem, we have the complete analysis. So this is final data. And we found that the phospho-tau 181 correlated with the NIA-B, which is the BRAC, tau, it makes sense, and also the ABC score, the IDNSC, the complete score. So it seems that phospho-tau is better for tracking the disease. Definitely than the amyloid markers, and also the neurodegenerative markers, the NFL and the total tau, which didn't correlate at all with any of the ABC stages. Okay, and to finish, the correlation with the non-AD pathology, the co-pathology. This is very interesting study because there is no data, and I will focus only on GFAP because we do not have completed the analysis, but GFAP is probably the most interesting because there is no previous data correlating with neuropathology. This is just to give you an idea. This is a correlation matrix between these are the blood biomarkers, and these are all the neuropathological variables that Alberto is collecting. You cannot read from there, but this is GFAP. I will only focus on GFAP. GFAP correlates with all the Alzheimer's disease variables, but also correlates with other neuropathology like hippocampal sclerosis, like TDP, or like Lewy bodies. We will see this in detail before. And just wanted to mention that these two variables that are in red are vascular variables. Alberto was saying how he's trying to score vascular changes, and vascular changes do not correlate with any of the blood biomarkers. This is very important information for clinicians because sometimes we have blood biomarkers results, we find vascular, and we don't know how these correlate, but we didn't find any correlation in our sample. So, this is GFAP. As I said to you, we will only focus on GFAP. GFAP, this is the univariate analysis adjusting by, multivariate analysis adjusting by covariates, but not by Alzheimer's disease pathology. So Lewy body, as you saw in the correlation matrix, is correlated to GFAP. So there are differences between individuals with positive Lewy body and negative Lewy body in terms of GFAP. Also hippocampal sclerosis, the same, and TDP. TDP positive versus TDP negative. But when you enter in the model the ABC score, which is the Alzheimer's disease pathology, we do this to see if this correlation is independent or not of Alzheimer's disease. We find that Lewy body is not independently associated with GFAP. If you enter Alzheimer's disease, the same with hippocampal sclerosis, but TDP43, it is independently associated to GFAP in spite of Alzheimer's disease. Okay, and this is the final slide. As a summary, we see that the Simoa blood biomarkers show excellent discrimination in our population, so pathologically confirmed late-stage dementia and cognitively healthy controls that has been stable for at least 10 years. A-beta 42/40 ratio, GFAP and NFL show the highest area under the curve, all excellent, more than 90%. So it seems that they are useful in diagnosis. It can help us to discriminate between cases and controls very well. I have to say that the phospho-tau 181 data is still not completed. GFAP and to a lesser degree phospho-tau 181 increase linearly until the premortem visit. So it seems to be the most useful markers for follow-up, for tracking the disease progression. Also GFAP and phospho-tau 181 were the only markers that robustly correlate with neuropathological AD rating scale. So again, that tells us that they could be useful for follow-up, for tracking how the disease changed. And GFAP was the only biomarker correlated with brain weight. So it's supporting the idea that this marker is very well correlated with disease progression until the very last stages of the disease. And GFAP was the only biomarker that correlated with temporal atrophy. We found this very interesting, pointing towards a special sensitivity of this marker to temporal lobe degeneration. And again, you have to remember that it was independently correlated to TDP43. That's, as you know, is a pathology that is it's targeting specifically, not specifically, but very intensively the temporal lobe. And that was independent of Alzheimer's disease pathology. And last, a negative result, but interesting result, is that the non-Simoa biomarkers correlated with vascular pathology. And I finished with the work in prose, which is a lot. We have a lot to do, a lot of data, very interesting. We have to complete the phospho-tau 181 in the Alzheimer's study to see performance and trajectory. This we need to be done. We have to complete the mixed pathology correlation with the premortem sample. This is a complicated analysis because all these variables are correlated, so we need to find ways to understand what is going on. And we are in the process of digitalization of the pathological variants. We are digitalizing the neuropathological preparation, and Ivan is doing using programs to quantify the pathological lesions. Will able to convert these semi-quantitative variables in continuous variables that will be very useful to correlate with biomarkers. It's going to be a very interesting thing to do. We have to complete the SIMOA determination in all the Vallecas cohort. At least we want to test 3 sample points to establish the trajectory also in the Vallecas. It will be very, very important to see preclinical trajectories. And we want to establish also to correlate, the biomarker trajectories with the different phenotypic information, with the brain imaging, with the neurocognitive trajectory, and also with genomics of proteomics that we also have for this cohort. And I finish acknowledging first all the patients of the Vallecas, both Vallecas study, our funders, and of course the Fundación team, the Fundación CIEN team. This is a very big study, and it's a teamwork, but especially would like to mention the person that have been more involved in this analysis, Diego Villalón. The biomarkers have been performed by Miguel Medina and Alicia Ruiz, and of course the Brain Bank, Alberto Rábano and Iván Burgueño, and thank you very much.
Thank you very much, Pascual. There are some questions over there.
Yeah. Congratulations, Pascual. This is a gigantic project and an enormous amount of work behind it. I just said congratulations, so I'm here. So, and very, very glad to see that GFAP is the best marker for most neurodegenerative diseases. And I think there is biology behind that, and one has to look to the paper so that we know why. So, but I have a question. So I didn't read well the slide with the cohort, but I got some part of the information, and I saw that 83% of the people recruited are women. So I wonder whether when you segregate gender, you get similar results or identical results. I think this is an important point. Thank you.
Yeah, you're right. That's a constant. In all these cohorts, there's a predominance of women. In the Alzheimer cohort, it's almost 85%, so it's overwhelming. In the Vallecas cohort, it's also very high. It's more than 65%. But all these analyses are adjusted by age, by gender, sorry. They are all adjusted. All the models are adjusted by gender, so we do not find...
Yes, I saw that in all the slides, but I don't quite well understand that. So when you segregate both genders separately, you get the same graphs, the same results. And so I would have liked to see gender, women and men separate in different
You mean to do a stratified analysis? A stratified analysis, men and women.
Yeah,
we didn't do this analysis, but the fact that, I mean, when you adjust in the model, you are taking into account differences. But I agree with you because these are a bit unbalanced because there are more females in the Alzheimer project than in the Vallecas, and it could be difficult to adjust when there's such a big difference. So it's a good, I think it's a good suggestion. I will do a stratified analysis to see more clearly where there is a difference. Thank you very much.
Great job. Congratulations, Pascual. I think one characteristic of your sample is the short time between the last sampling or serum sampling and the exitus. And in this sense, did you check if several markers or combination of biomarkers has predictive value about over exitus of the patients in temporal terms?
You mean if they affect survival? If the markers affect survival?
Absolutely.
Yeah, Yeah, that's very, very good suggestion. We didn't do that, but we will definitely. Yeah, that's a very good suggestion. Thank you. Yeah.
I have a question here.
Very interesting. Me? Okay. I'm curious to see that some of the most reliable biomarkers are intermediate filament proteins. I was wondering whether, has anybody checked the biophysical properties of these molecules that may determine or condition their stability in blood, so they become reliable markers because there is something that makes them more stable or with respect to other markers. And in regards to that, maybe vascular pathology could take that into account for the intermediate filaments of endothelial cells.
I don't know if I understood you well. I think you mean if there are conditions that are related to, different to pathology, but that are related to the... Yeah, I see. Yeah, that's a very good question. Yeah. That's, I don't know. I'm not sure if this has been taken into account. That's a great question. I mean, I know that there is a group that is analyzing all the conditions that affect, like freezing, thawing, and well, also the conditions in the body that may affect the renal, kidney function, the liver, and some of these things affect biomarkers, especially NFL. I thought you were going to ask me for this increase in NFL that we find in the last stage of the disease, which is difficult. Yeah. And, all these things could, but the thing is that the field is very young. So this is just 2 years ago that this started really becoming more frequent. So we don't know. There are many questions. I just put some of these questions that we can address, but there are lots, of course. And what you're saying is very true. And also NFL is affected, for example, a lot by peripheral neuropathy. So this is one of the things that is hampering its clinical application, because peripheral neuropathy is very common in old people. And my guess is that this increase that you find at the end is that people is in bed, and when they are terminal, most of them they are in bed, so probably they get more NFL, peripheral NFL, because it's such a difference between the trajectories. So I don't know. That's, of course, a speculation. But,
Hi. I have a very general question. Congratulations for the study. I just wonder why plasma and not serum, for example. Did you ever check serum? I mean, I see that most of the time is plasma biomarkers, and I wonder whether is there a particular reason why people focus on plasma and not serum?
Well,
and also about, sorry, just to finish the question, so about plasma protein isolation conditions, I don't know how it works, the protocol, if you have to centrifuge once or twice, if that makes it makes a point in the results.
I didn't catch the last part of the question.
The question is, why plasma, not serum?
I got
and
that.
With plasma, do you have to centrifuge the plasma, the blood, once or twice, or does it make a difference in the results? Did you check that?
Okay, these things. Yes, I mean, that's a question more for Miguel. That I mean, the serum or plasma is the kit. I mean, the CIMOA tells you which.
I don't listen very well. Can you please put the micro in?
Sorry.
Yeah. Sorry.
I mean, we just follow the instruction of the quantity. Some of these markers are validated for plasma or both or serum. So we just follow what they say, why it is validated, and how is centrifuge. I mean. I don't know, maybe Miguel can answer that, and maybe in the coffee later. But this, as I mentioned to you, this is very new field, and there is a consortium trying to answer this question. There are some publications about all the management of the samples and how they affect the results, which are very, very important point, definitely.
Thank you very much. I have a sort of philosophical question, a bit provocative, so please don't get upset with me, okay?
Oh.
So CIMOA is a wonderful tool, and I think at this time, you go to PubMed, there are over 500 papers working on CIMOA, and all the people who have a bit of money and can buy this equipment are working on this. So now we have a very nice window to analyze, for diagnosis mainly, with the multiplex system that they have, A-beta, phospho-tau, whatever, and GFP as well. So at one point, what we are doing is just to characterize very nicely our cohort, but we are not having an increase in the research, in research on Alzheimer's disease. Again, my point, sorry, I have to insist. My point is that we need transcriptomics. Circular RNA, microRNAs, coding RNA. So just get the packaging tubes.
Okay. I friendly disagree absolutely to that. I mean, I think this is research that is very important for us. It's clinical research, of course. It's not mechanistic research. We are not going to discover the cause of these things, but I don't care. I mean, if I have something that predicts very well how my patient is going to develop and is useful for me to diagnose and follow up, that's what I want. And it's very important, believe me, to how you manage the patients, and that is very valid. Of course, we are also doing, I agree with you, it's essential to understand and to get even better biomarkers. I mean, as Alberto mentioned, there is a lot of complexity there, and I think just GFAP, tau, and amyloid beta probably will be not enough if we want to really see the granularity of our patients. And actually, okay, both. Actually, we are doing proteomics also in our sample, which I think is going to be great. We are doing the Olink 3K. It actually has been done. We have the results. We have to analyze, but we have many work in progress, as you see. And we are going to now to correlate the proteomics with the neuropathology. Ideally, quantify neuropathology. We are digitalizing. And also with the genomics, with the GWAS. So we are also with you. I think that this is an important thing to do.
Thanks, Pascual. I think we have to close down because we run out of time. So I'd like to thank all the speakers of this session for their presentations. And we have the coffee break. We'll be back here at 11:30.